Very good afternoon to one and all. I welcome you all for the session 14 of 5 days 3D printing FD. So for this session, we are having Dr. Ruben Rex Peter, who is currently working as a research scientist at Beatson Institute of Cancer Research, Glasgow. He is currently working with the control of cancer cells. Before to that, he is associated with Cardiff University, UK as a postdoctoral research associate. He received his PhD in medical science from Warwick Medical School, University of Warwick, Coventry, UK, and his master's degree in M Research in Biotechnology from Anna University, Chennai, India. And he received his master's in science in Applied Biomolecular Technology from University of Nottingham, UK. He received merit scholarship for excellence in academic performance for 3 consecutive years during 2002 to 2005 at Vellore Institute of Technology, Vellore. He has published more than 10 plus research articles in original SCA and Scopus Index journal. His area of research includes RNA DNA extraction, cDNA synthesis, primer design, cell metabolic analysis using seahorse, functional assay using cell ligand, and flow cytometry analysis. I request Dr. Ruben to take over the session.
Hello, hope you all can hear me.
Yes, sir.
Yeah. Hello, good afternoon, everyone. As Dr. Hariharan mentioned, I'm Ruben. I'm going to be presenting this session with you guys. I first of all would like to thank Professor Rajendran for giving me this opportunity to talk in this FTP program, and Dr. Hariharan for making this happen. So as you all know, I'm going to talk to you about in vitro analysis of additive manufacturing component. So I just wanted to give you a brief introduction about my experience in 3D culture of cells. I have near about 13 years of experience in this area. So this started with when I was working at a company in Chennai. It's called NCRM. So we used hydrogel, which is a gel-like structure for culturing stem cells. So this helps stem cells to body-like condition. Next, during my PhD at University of Warwick, I have created a 3D human endometrium, which is a female reproductive organ. So this could be used as a tool for testing embryo implantation before women go into IVF. And then during my postdoc research experience in Cardiff, I have created a 3D structure to study tumor and immune cell interaction. So this is quite hard to nail. So I have created a tool which could be used as a easy manipulative tool for accessing tumor cells and how it interacts with immune cells. Now, currently, I'm working at Beatson Institute. So I'm working on colorectal cancer. So these are some of the organoids I'm currently growing in matrigel, which is like a soft gel. And if you, I think you can see that intestinal kind of structure are growing in the matrigel. Coming into additive manufactured products in biology, where they use something called bioink, and it's used for making hard material as well as soft material. Hard material, I am sure you guys are experienced and experts in, which I have very less knowledge about. But anyhow, for creating a hard material, they use they construct the scaffolds, and cells are cultured and seeded onto the scaffold, and then it is implanted into the patient for regeneration of tissues. Whereas in case of soft material, either it can be a gel or it can be a harvested tissue, and it will be normally decellularized if it's from it can be from human, or sometimes it can, like heart valves can be obtained from pigs even, and they support like human heart cell culture, and they will be mixed with hydrogel, as I have mentioned so far, and they will be implanted into the humans, and it can be used in different varieties. The ultimate goal is to create an environment like human body, which is in a 3D architecture. So what is an ideal scaffold in a point of view of an biologist? The scaffold should allow cell adhesion, maintain cell viability, promote cell growth, allow the retention of differentiated cell functions. The material should be biocompatible. It should not provoke any inflammation or any toxicity, which is obvious thing. It should be biodegradable and be eliminated eventually. Even if it's not biodegradable, at least it should integrate with the actual existing structure. For example, if we are using a scaffold for a bone structure, it should integrate with the bone, and it should not be looked as a foreign by the immune system. That's the main purpose. Porosity should be there to provide sufficient space for adhesion, ECM generation, and providing diffusion of nutrients during culture or even in a body condition. The material processing should not affect its consistency and should be mechanically strong. So what is bone? Bone is a specialized connective tissue with cells and mineralized substances. Organic components of this bone is osteoid, something called osteoid. Inorganic component is the hydroxyapatite. The ground substance is non-collagenous glycoproteins. And in our body, we have flat bones and long bones. Flat bones, a typical example is skull, which is developed during your developmental stage, like when you are in your mother's womb. So during that time, what happens is flat bones are created by a method called intramembranous ossification. This is where mesenchymal stem cells, it's a kind of stem cells. Stem cells are what you can consider a stem cell like a branch of a plant. If you stick a plant somewhere, it will grow. So stem cell is a kind of, you can take that as an example. So mesenchymal stem cells condense within highly vascularized embryonic connective tissue and transform into osteoblast. In case of long bones, example femur, it's formed by something called endocontrol ossification. This endocontrol ossification is a way of body creating a strong bone. So here also the mesenchymal stem cells differentiate into chondroblast. Cartilage bones will be formed first, and then later it will be replaced by a bone tissue. So the chondroblast kind of form a mold for the bone cells to create. After that, the chondroblast will be eaten away by immune system, and then it will be replaced by bone cells. There is homeostasis between osteoblast and osteoclast. These are like different type of cells which maintain the good bone nature in a bone. So osteoclast basically eats away the bone, and osteoblast kind of regenerates the bone. Bone fracture and fracture healing. There is a different type of bone fracture. I don't want to go deep into medical terms, but anyhow, if you consider a bone fracture, it can be complete or incomplete bone fracture. Complete in the sense that a section of bone completely miss from the structure of the bone, whereas the incomplete is like shredding of bones here and there. It's not a complete fracture. So how it starts to heal? It happens in 3 different phases: reactive phase, reparative phase, and a remodeling phase. So I'm going to tell you in detail how this happens. So there will be a form of blood clot, which is termed as hematoma, due to the bleeding at the fracture site. And this is the first stage of fracture healing. Then blood vessels starts to form within this blood clot. So blood clot is a good thing. So blood vessels starts to form within this blood clot. After that, phagocytes, which are immune cells, a type of immune cells, their main role is to eat away any foreign material or any unwanted material in the body. So what they do, they come into the via the blood vessels and they remove the non-viable material. Fibroblasts, they also come. These fibroblasts are basically skin cells. They also come via the newly formed blood vessels, and they lay down like collagen monomers to form a bone matrix. Calcium hydroxyapatite is deposited. This is a inorganic material which I mentioned before, in the form of insoluble crystals. So this process is called mineralization. So mineralization of collagen matrix hardens it and transforms it into a bone. So that's basically how the fracture starts to heal. The cells which are involved in this process are mesenchymal stem cells, chondrocytes, which are basic cells of cartilage, endothelial cells, which are blood cells. Talking about biomaterial scaffold that can be used for bone tissue engineering, the ideal scaffold should be osteoconductive, osteoinductive, and support osteogenesis. What do I mean by osteoconductive? It should support bone cells to survive, basically. Osteoinductive, even if there are stem cells, it should be able to induce those stem cells to form into a bone cells. And support osteogenesis, it should support the process which I mentioned before, like it should allow blood cells to come in, to allow the phagocytes to eat away any unwanted material on the scaffold. So it should basically support whatever I have mentioned in the previous slide. So the composition of scaffold, their geometrical form, surface character, and internal architecture like pore size, porosity, and pore structure are crucial for their osteoconductive properties. There are different types of scaffolds available. Biological scaffolds, basically corals, collagen type 1, demineralized bone matrix. There are synthetic scaffolds, which are porous metals, ceramics, bioglass, calcium carbonate, and sulfate. And for the project that I have worked on, the material, polyamide material, was provided by Dr. Hariharan, then Mr. Hariharan. So he provided me with the polyamide for this study. So what is a polyamide? Polyamide is a structure, as the name suggests, it is formed by a repetitive sequences which are connected by an amide linkage. There are naturally occurring polyamide. The best example are wool and silk, and there are like artificial polyamides, example nylon, those kind of things. The artificial polyamides are created by step growth polymerization or solid phase synthesis. And the material which we used for this study was P82210FR. So this polyamide material is hardened to replace missing bones using a rapid prototyping method. So it can be customized to suit patient needs. So it is a fine polyamide. It is halogen-free, and it has good mechanical properties. It has excellent long-term constant behavior. It's fully functional, load-bearing plastic parts of highest quality. The elements which are in this polyamide are there is carbon, there is oxygen, there is other materials, inorganic materials. So basically, there is nothing which can be considered as toxic. And the whole idea was this polyamide material will be used in the place of a missing bone, and it should regenerate bone growth. So my aim was to test whether this customizable polyamide scaffold is biocompatible. So I used mesenchymal stem cells and other cell types. So I have used different type of cell types. And you need to remember that the cells I have mentioned so far, like mesenchymal stem cells, bone cells, or endothelial cells, these are the different cell types I've used in order to check whether this material is biocompatible or not. So the cell types are mesenchymal stem cells. So these are fat-derived cells. These are plastic adherents, and they can be differentiated into osteoblasts, which is the bone cells, or it can be even differentiated into fat cells. I have used chondrocytes, which are obtained from the patients, and EA-high 926, which is a endothelial cell line or a blood cell line. And then I have used something called SAOS2, which is osteosarcoma, basically a bone cancer cell line, but it can behave in a similar way as a bone cell. So it can be used in the place of, like, it is hard to obtain bone cells and culture in lab conditions. So I have used like a bone cancer cell line, which is a replication of bone cells. And I have used something called MG63. This is another bone cancer cell line. However, this is similar to mesenchymal stem cells. So this can be differentiated into bone cells, but typically it will behave like a stem cell. So the markers for a bone cells won't be present in this MG63 unless or until it is differentiated. So what all I have studied with the methods for studying the biocompatibility. So I have analyzed adherence, viability using live data analysis, the ability to support bone cells. I have used a testing called alkaline phosphatase staining, checked proliferation using something called MTS assay, and I have checked whether it will support differentiation using osteogenic induction medium. So for adherence, what I have done is I used the polyamide scaffold, seeded 10,000 cells, and then cultured them for up to day 14. And then I have counted the cells on day 1, 7, and 14 to find out how many cells are adhered to the scaffold. So for this, I have used this calculation. So non-adherent cell ratio is 1 minus non-adherent cell number divided by the total number of cells times 100. So the number of non-adherent cells is basically the cells floating in the medium, and the adherent cells is the cells. So you will this is like an indirect counting of the adherent cells on the bone, on the scaffold. So in case of SAOS2, which is similar to bone tissue, you can see that it is adhered. Adherence is increasing, like from day 1 to 14, and it is near about 100% in the case. In case of MG63, which is again similar to stem cells, but has quality into differentiating into bone cells. So they are also adhering better. So they are also near about 100%. The stem cells, they are adhering in a less numbers compared to the bone cells. Whereas chondrocytes, they start very slow, whereas on day 14, they also reach to near about 100%. Endothelial cells, the blood cells, they are able to increase as well. So initially, maybe these cells are having issues sticking to the scaffold. However, over time, they are able to adjust themselves to grow on this scaffold. Then moving on to viability study. For this, I have used double staining kit, which is a regular type of kit used in biological labs. So this kit has something called calcine AM and propydium iodide. So calcine AM, what it does is it will turn into a green color, and this can be done only by a viable cell. Whereas if it's a dying cell, the disruptor membranes will be incorporated with the ethidium homodimer, which is the propydium iodide, which I have mentioned, and it will turn the cells into red color. Basically, green means live, red means dead cells. So this is the study design. Cells used for this assay are mesenchymal stem cells, chondrocytes, bone cancer lines and the endothelial cells. So 10,000 cells were seeded per well containing the scaffold in triplicate. They were incubated and assay was carried out on day 1, day 7, and day 14. So the calcium and PI, propedium iodide, were added to the media and incubated for 30 minutes. And then it was visualized under a fluorescent microscope. And we have chosen like 4 different zones on the scaffold and then counted the green and red cells basically. So as I mentioned, so the green means live, red means dead. So I'm showing these representative pictures of mesenchymal stem cells. So you can see there are loads of red color on the scaffold. I'm not quite sure whether you can see the red staining. There are 1 or 2 cells here and there. So I've done the counting. So I'm going to tell you the results, what they look like. So this is the live dead of mesenchymal stem cells on the scaffold. So how this is calculated is we count the number of green cells and then divide by the total number of cells seeded and then times by 100 to get the percentage. So same for the dead cells. So here you can see that there are more live cells compared to the dead cells in case of mesenchymal stem cells, and it's maintained from day 1 to day 14, which is a good thing. And then in case of chondrocyte, again, there are more viable cells compared to dead cells. And towards day 14, you can see that it's going near to 0. Like the chondrocytes are able to keep their viability on the scaffold in a better way. And this is for SAOS2, which is the bone cancer cell line, or similar to bone tissue, that it is also maintained with good viability. So this MG63, which is similar to the mesenchymal stem cells, so they are having more number of dead cells on day 1. However, towards day 14, the number of dead cells go down. This is for the endothelial cells. They are also maintained in good level. So from this, what we can see is the scaffold is able to support the viability. It's not having any toxic activity or any unwanted reaction to the cells. So the cells are able to survive. The cells needed for the bone regeneration, such as mesenchymal stem cells, chondrocytes, blood cells, and even the bone cells themselves, they are able to survive on this scaffold. So which is good news. So to check the ability to support bone cells, and I did something called alkaline phosphatase activity. So again, this was performed on day 1, 7, and 14. So for this, we use something called fast-red TR and stable naphthalate phosphate. And this was done mainly on the bone cell and the MG63. As I mentioned, if you remember, the bone cells will have this alkaline phosphatase. In human body, the 2 main things that express this alkaline phosphatase is one, the major organ is the liver, the other organ is the bone. So if it stains for alkaline phosphatase, that means we are staining the bone cells basically. So this is the study design. Again, cells used for this assay is SAOS2 MG63. 10,000 cells were seeded per well containing scaffold in triplicate. So incubated with ALP staining assay and carried out on day 1, day 7, and day 14. And then it was visualized under a fluorescent microscope. So these are the representative images. So what I've done is I have imaged the cells on the plate, which is a 2D culture as well, and cells on the scaffold. So this is the day 1 image. So this was imaged at 4 times under a microscope, fluorescent microscope. The red color means it's positive for alkaline phosphatase activity. So on day 1, you can see there is only less number of alkaline positive bone cells on the scaffold, whereas it increases upon day 7, and day 14 it is the scaffold was completely filled with alkaline phosphatase positive bone cells. So this using the the bone cells, SAO-S2 cells. Whereas in the plate, you can it is differentiating. It is not maintaining the structure of. If you look closely, the cells are differentiating much, like they are getting older. Whereas on the scaffold, it's maintaining its structure, and it's maintaining good. So this is the negative control using the MG63. As I mentioned, it should not behave like a bone cell. It is basically a stem cell. So there was no alkaline phosphatase activity. So it is not doing anything. Then we wanted to check whether the scaffold will support differentiation. So mesenchymal stem cells on the scaffold and plastic, which means basically a 2D cell culture plate for 14 days in osteogenic medium, was changed every day. So osteogenic medium contains these chemicals: glycerophosphate, dexamethasone, ascorbic acid, and then alkaline phosphatase activity was carried out towards the end of the study. So this is the picture which was taken on day 14. These are the cells on plate, and these are the cells on scaffold. So you can say we can say from this that the scaffold is supporting differentiation, like when the correct chemical, or if you think about the body condition, if there is like correct situation, the scaffold will support bone differentiation from the stem cells, which is a major important characteristic needed for a scaffold. So this is the proliferation assay. This was using MTS assay. So basically, this is similar to the calcine assay, which I mentioned. Only the viable cells or multiplying cells will be able to reduce a chemical tetrazoline compound in this assay to form a colored substance. So the color can be measured colorimetrically, and you can say if it's increasing, you can say this many numbers of cells are proliferating in this assay. So assay design, 10,000 cells were seeded per well, containing the scaffold in triplicate, incubated for 24 hours. And we need a standard, because it's a colorimetric assay, we need a standard to extrapolate our results to find out how many number of cells are there. So incubated for 3 hours, then PMS and MTS were added in equal amounts, and then incubated for 1 hour, and then they were read at 492 nanometer and 650 nanometer for 4 hours. So this is the results. So comparing day 1 and day 7. So on day 1, there were 10,000 cells in case of the endothelial cells, and on day 7, it is proliferated up to 30,000 cells, which is 3 times increase. And in case of endothelial cells, on day 1, there were 8,000 cells, and on day 7, it is increasing, but not in a massive number. So this is also good news, because endothelial cells need to be there, but they don't need to multiply or anything on the scaffold. So it is increasing to an extent which is supporting our hypothesis that this will support the bone regeneration. Moving on to surface of the scaffold. This was done using a confocal microscopy. Again, this study design scaffold was incubated in rhodamine dye. So the whole idea behind the study was the rhodamine dye would color the scaffold red, and we would have the cells as in green color, but that didn't happen. So I'll show you the results. Washed with PBS to remove the excess dye, and then cells were seeded at 10,000 cells per scaffold, and then they were incubated for 24 hours. So calcium dye was added and incubated for 30 minutes, and it was visualized under confocal microscope at 480 nanometer and 495 nanometer, which are red and green color. So we expected the scaffold to take up the red color and the cells would appear green color, whereas the cells also took the rhodamine dye, and they got double stained, and the scaffold wasn't dyed at all or stained at all. So we can see that cells are growing fine. These are mesenchymal stem cells. They are growing fine on the stems on the scaffold. So this is the mere scaffold, and this is the overlay of red and green color. So altogether, my observation from this study are polyamide scaffold supports adhesion, proliferation, differentiation, and viability. It supports osteoblast kind of cells, osteosarcoma, because we used osteosarcoma cells. Scaffold supports most adherent cell types. Those are involved in bone regeneration. It supports differentiation of mesenchymal stem cells as showed by alkaline phosphatase activity on day 14, which shows that it's osteoconductive. Live-dead assay shows that mostly viable cells. MTS assay shows increase in number of cells in all 3 cell lines, in particular endothelial cells where there was a really good increase. And from the confocal study, you can see that there are no pores and the cells are growing flat. So suggestions, like from our working point of view, to make our lives easier in the lab, the scaffold to be porous or thinner would be easier for us to handle. To use hydroxyapatite coating to make the scaffold osteoinductive. To proceed with animal studies to generate some interesting data. And I would like to thank my PI at the time, Dr. Anuradha Dhanasekaran, and manufacturing department of Anna University, Dr. Arime Kannu and Mr. P. Hariharan, and Aston University at Birmingham, UK, where I have done most of the experiments which I have showed you the results today. And this was helped by Professor Eustace Johnson, and their mechanical department were very helpful in making the scaffold quite thinner to make it usable in the lab. And thank you. This beautiful picture of Glasgow at night. Thank you. Are there any questions? I'm happy to answer.
Dear participants, you can ask any questions related to the presentation. Since this is very short presentation, it is mostly related to the in vitro analysis, how additive manufacturing components are reacting with the life science. Right? So you can also ask related to life science related questions. What kind of cell line studies has to be done for any additive manufacturing components? That's kind of a thing.
I feel that it's quite important. Like this needs to be considered if you are thinking of a material which can be used in a patient situation or in a hospital situation. I think these needs to be considered. Like rather than maybe you guys are coming from a mechanical point of view, whereas we would be able to suggest something in a biological way.
Yes, sir. So if no more questions, we shall close the session, right? So I would like to thank Dr. Ruben Rix for the wonderful session. Have given the insight on in vitro analysis, what kind of a cell line studies has to be done in the additive manufacturing component to substantiate that product to be used in the biological environment. And thank you participants. We shall join for the next section by 2:30 p.m.